let's officially start our meeting. So first, I would like to thank you again, Florian, to accept our invitation. For us, it's a great honor to be here with you and also share this hot topic on plant physiology. I believe that it will be very great for all of us. And I also would like to thank for all the people that join in this meeting of LabPlantTalks. And now I will pass the words for Florian, and also to introduce a little bit him. Florian now is a lecturer on the University of Birmingham on UK. And today he will talk with us about the photorespiration. And now I will pass the words for him to explain for us with more details. So Florian, thank you again, and be free to start your seminar.
Yeah, thanks for inviting me. This is exciting to be able to present all the way in Brazil, even though I'm not even moving. But yeah, it's a great opportunity to present my work, and I'm glad all of you are showing up today. Let me just see if I can share my screen. So I hope you all can see my title slide.
Yes, it's working.
Excellent. Okay. So today I'm going to talk about photorespiration, which is sort of the overarching theme where pretty much everything I've done over the last 10 years or so sort of fits in. And this is sort of an outline of a review that I published earlier this year on photorespiration, and sort of some ideas that I wrote down in this review. So yeah, it's all about photorespiration. And the question is photorespiration a good thing or a bad thing for the plant? And maybe I can sort of discuss this a little bit further in the next 45 minutes or so. So there we go. Okay. So in the light, plants take up CO2 and fix CO2. This is what we usually call photosynthesis. But at the same time, the plant also releases CO2, both through respiration, which we usually call RD here, respiration, and also photorespiration. So there's these two, CO2 uptake and CO2 release, happening at the same time. And the net CO2 uptake, so the assimilation rate of the plant, we can therefore describe as, so net assimilation rate A, as the difference between the carbon taken up from carboxylation and the carbon released from photorespiration and respiration. So that's sort of a simple way of quantifying how much carbon a plant can take up at any given time. And as this equation outlines quite nicely, is if you release more CO2 from photorespiration, then that also means that your net assimilation rate is becoming smaller, and the plant is sort of losing out on photosynthesis, on CO2 uptake. And so the question is, how big is this impact of photorespiration on the net assimilation rate of the plant? And Walker et al. Came up with a paper a few years ago where they try to quantify the impact of photorespiration on plant biomass, or in particular of the yield in soybeans. So they've done this for every county in the US here where they grow soybeans. And then they've modeled the impact of photorespiration that it has on crop yield. And as you can see, the loss is sort of in the range of sort of 35 to 60%, depending on where you are. And you can also see that further in the south, you get a higher loss in yield just because it's warmer and photorespiration goes up during temperature. But before I go into detail about the impacts, let's maybe start out with this maybe a bit more complicated slide that we spent a few minutes on to see what photorespiration actually is, or what you might consider photorespiration to be. So you might see it as everything starts at Rubisco. So Rubisco is the central enzyme driving photosynthesis, and it uses this RUBP as its substrate, and then carboxylates that. So it takes the CO2 from the air and sticks it on to produce 2 molecules of phosphoglycerate. And this phosphoglycerate is then metabolized in the Calvin-Benson cycle back to RUBP. And the extra carbon that is added here is then ultimately diverted from the Calvin-Benson cycle as triphosphate. And that's really the carbon that the plant assimilates. But Rubisco has this curious property that it can also use oxygen as a substrate. So instead of attaching a CO2 to the RUBP, it can attach an oxygen and then produce only 1 molecule of phosphoglycerate and 1 molecule of phosphoglycolate. And this phosphoglycolate is toxic to the plant, so it needs to be metabolized fairly quickly. And that's what this pathway down here is good for. So phosphoglycerate is first, phosphoglycolate is first converted to glycolate, glyoxylate, and to glycine, involving both the peroxisome and the mitochondria as compartments in the plant. And then converted to serine and back towards glycerate and phosphoglycerate. And so this phosphoglycerate enters the Calvin-Benson cycle again. So there's one part that's important, which is the stoichiometry. So you have 2 molecules of phosphoglycolate make 1 molecule of phosphoglycerate. And that also means you have 4 carbon atoms here that make up those 2 molecules that get converted to 3 carbon atoms on this side. And that also means that in the conversion from glycine to serine, there's 1 CO2 that's being released. And this is what we call photorespiration, or the true meaning of photorespiration. So that's the CO2 that the plant will give up. And this is sort of what goes into this equation here as the CO2 being lost. The other thing that I should probably point out at this point is that in the conversion from glyoxylate to serine, we go from a carbon-containing metabolite to a carbon and nitrogen-containing metabolite, like this amino acid. So we have to stick some nitrogen in here. That is then taken away again in the conversion from glycine and serine as ammonia, or in the conversion from serine to hydroxypyruvate, which also produces glycine. So there's this nitrogen pathway that sort of supports this conversion from glyoxylate to amino acids and then back to hydroxypyruvate. So there's a tight link between photorespiration and nitrogen assimilation, or nitrogen metabolism, let's say. So so the true sense of the word photorespiration is this CO2 being released. But you can also see it as this pathway here that recycles phosphoglycolate. So the photorespiratory pathway really is the metabolite pathway that involves all these enzymes here that make this happen. And people have been looking for those enzymes and describing those enzymes for quite a while. And they have all these one characteristic property, which is if you knock them out, or if you produce antisense plants to those plants, when you grow them in high CO2, like 1 or 2% CO2, they pretty much grow the same as your wild-type plants. But if you shift them to ambient CO2 concentration, so let's say 400 ppm, you can see that many of them, or most of them, have a severe growth phenotype. So they do very badly when there's a lot of oxygen relative to the CO2 concentration in the air. So, and the reason is because Rubisco sort of has this dual substrate, CO2 and oxygen. And that's sort of the third way we can describe photorespiration is what happens to Rubisco. So Rubisco, in its inactive form, has to bind an REBP first to become an active Rubisco enzymatic site. And then it can either react with a CO2 or with an oxygen, and then go one way or another way. But really, what determines how much, what the rate is to carboxylate this active enzyme versus oxygenate this active enzyme depends very much on the Rubisco enzymatic properties, in particular Rubisco specificity for CO2. So how good can it distinguish CO2 from oxygen? And the second important parameter is the CO2 concentration relative to the oxygen concentration. So if there's more CO2 around, then obviously that reaction will be favored relative to the reaction with oxygen. So we really need to know the ratio of CO2 to oxygen at the site of Rubisco, at the active site of Rubisco. And because oxygen is so much higher in concentration than CO2, we can take it pretty much constant. So really the only important parameter is the CO2 concentration. So these are sort of several ways what you can see of as photorespiration, and they all have their sort of validity. So we can mathematically quantify it or describe the effect of photorespiration on the assimilation rate. And Farquhar et al. First did this with this equation here, or one similar to that, where they've said, well, we can take our assimilation rate and we can describe it as the minimum carboxylation rate that you could get under the three limitations that exist in the plant. So one would be Rubisco limitation. So depending on how much enzyme and how fast that enzyme can take over CO2, you get a certain rate. Or the electron transport limited carboxylation rate, which depends on essentially how much light the plant can absorb and convert into energy to regenerate REBP. And then the third limitation would be triphosphate utilization. So how much of that fixed carbon can the plant actually metabolize? So Farquhar et al. Described this assimilation rate as the minimum of these rates. And then you account for photorespiration and respiration down here. So photorespiration, the part here, this gamma star parameter, this is where the specificity of Rubisco for CO2 goes in. So that parameter describes how well Rubisco can distinguish between CO2 and oxygen. And then down here, C, that's the chloroplastic CO2 concentration. So this tells how much CO2 is actually inside the chloroplast where Rubisco is. So in the end, we have the carboxylation using of CO2 versus the oxygenation. And the rate of these two will depend on specificity and also on the concentrations of those two gases. So if we say oxygen is constant, then as we change CO2 concentration, we will change how much Rubisco can react with CO2. So as we change CO2 concentration, we should be changing the rate of CO2 uptake. And when we measure that, we usually get something like that. So we have an initial increase in assimilation rate that sort of starts to flatten out as you increase the CO2 concentration. And at low CO2 concentrations, you tend to be Rubisco limited. So this is the range where as you give more CO2, or if you have more active Rubisco, you would get an increase in CO2 uptake. And at slightly higher concentrations, that's where you become electron transport limited. So that's where the regeneration of RUBP is limiting CO2 uptake. And then even higher CO2 concentration, that's when the plant becomes triphosphate utilization limited. So the general pattern is always the same, but where the plant is limiting, or what is limiting photosynthesis in a plant at any given situation might change. So a few years ago, I've worked on this, trying to map out under which conditions you sort of get what limitation. So here I've plotted the assimilation rate of a plant relative to leaf temperature and CO2 concentration. And you can see that depending on where on the surface you are, you end up with the different limitations, which are color-coded in the same colors. So here in the black line, that would be your ambient CO2 concentration temperature response, going from low temperatures down here to high temperatures. And you can see that you tend to go from the yellow triphosphate utilization limitation towards, at low temperatures, towards Rubisco limitation at high temperatures. So really what is ultimately limiting depends on a lot of different parameters and components. Yeah, so one more thing is, maybe you've noticed that before. So as you go to higher and higher CO2 concentrations, often what you see is you get a decrease in CO2 assimilation rates. It's not strikingly much, but it's very common and noticeable. So relative to this straight line here, what you might assume to be triphosphate utilization limitation, you get this sort of hump on extra assimilation rate on top of that, which is sort of highest in intermediate CO2 concentrations. And that, it turns out, has to do with photorespiration. So the photorespiratory pathway produces a number of metabolites that are useful in other biochemical pathways. So in particular, those amino acids, glycine and serine, they can be taken out of the pathway and used for other things. So glycine and serine are used for protein synthesis. So that's the main pathway to produce those amino acids. So they can be taken out of the photorespiratory pathway. They're used to produce glycine betaine and glutathione, which are sort of stress response compounds. And then serine, for example, is also used for the sulfur metabolism, making cysteine. And in any of those cases, you take out ultimately an amino acid, glycine or serine. So if we take out an amino acid, we not just take out the carbon, we also take out the nitrogen off that part. That also means that nitrogen is not being fed back into this cycle here, but has to be replenished from a new nitrogen. So this is where the plant actually, or at least one of the ways where the plant can actually assimilate nitrogen from de novo. So that's where that pathway comes in. But we have also another compound here. So this is methyl tetrahydrofolate, which is the source of all one carbon compounds that are used to methylate any sort of biological biochemical reaction. So the plant uses this component to make chlorophyll, for example, or many secondary products, and particularly lignin for trees, or things like nicotine and tobacco, or betaine and beet. So a lot of carbon is diverted from the photorespiratory pathway into these compounds via this tetrahydrofolate. And conversely, that is sort of what describes this hump up here, and that's the carbon going into these compounds. So then the question is, if this is related to nitrogen assimilation, then we should be able to modify how much nitrogen we feed in here to see how much glycine and serine can be diverted away from the photorespiratory pathway. And I've tried this in a little experiment. I've taken sunflower plants, and I've measured sort of an ACI curve, so a CO2 response of the assimilation rate. And you can fit this model here, the solid straight line, to the data, and you come out with this value alpha, which tells us that 27% of the photorespiratory carbon is actually diverted away from the photorespiratory pathway. So 20% of the carbon coming down here is not being returned into the carbon-Benson cycle, but leaves as serine, glycine, or THF. So if you then compare that to, if you say, well, what would it be if this were to be 0? Then we get this dashed line here, and you can see, because we can divert carbon from the photorespiratory pathway, we actually get an increase in CO2 assimilation rate. So what happens when we change the nitrogen supply of the plant? So if I take those plants and I dump a bunch of nitrate onto those plants, we get those red curves coming up. So we see that CO2 assimilation rate, especially in that central region, increases. If we take the same amount of nitrogen and add it as ammonia, we also get an increase in assimilation rates, but it becomes much more flat up here. And the reason is, well, at least I think, ammonia is not really transported in the plant from the root to the leaves. So ammonia is mostly assimilated in the roots into amino acids, and then the plant transports amino acids instead. So the ammonia doesn't really have any opportunity to interact with photorespiration in the leaf. So, but what we're really interested in is, how big is that hump in the middle here? So if we scale this to sort of the trisphosphate utilization rate in the end, we get this here, and you can see clearly. So as we add nitrate, we increase this hump in the middle, and as we add ammonia, we decrease this hump. And you can also quantify that. So if you add ammonia, we get about a 4-fold increase in this fitted value alpha. So the proportion of carbon being taken away as serine and glycine. Whereas if we add ammonia, we get about an 80% reduction in that value. So that's a clear or nice evidence that photorespiration and nitrogen assimilation rate actually interact, and that photorespiration does something good with nitrogen assimilation rate, or something useful, let's say. So that also means if we take away photorespiration, we should have a similar effect on changing the amount of nitrogen supply. Right? If there's no photorespiration, then there shouldn't be any nitrogen being assimilated or any glycine serine being diverted. And so if we take those same plants and we measure the ACI curves at ambient oxygen concentration, so 21%, we get those orange dots. And if we take those same leaves and we measure that at essentially no oxygen concentration, so where there's no photorespiration or no oxygenation reaction happening, we get those blue dots. And you can see that assimilation rate is increasing here, whereas it's is decreasing here, where it's increasing in this area here. So we can then take the model from before and fit all this, and we can fit both curves with the same parameters, except for changing the oxygen concentration in the model. And we can see that it fits both. So the model saying, well, if you assume some carbon is being diverted through nitrogen assimilation, fits perfectly with the data that we collect. So from that, we can see that in this area here, where photorespiration is actually increasing CO2 assimilation rate, that's where we get this interaction being beneficial between photorespiration and nitrogen assimilation. So then that's when the plant creates essentially a second sink for carbon. So it doesn't assimilate directly only into carbohydrates, but also into amino acids, essentially. So first bottom line here is that photorespiration can actually be beneficial and increase CO2 uptake despite reducing the efficiency for the carboxylation reaction of Rubisco. Okay, so that was the first point. Now I want to go a little deeper into how we can model photorespiration and what else we can learn from that. So if we take our data that we've measured, again, an ACI response with photorespiration, so at normal levels of oxygen, and we compare that with the data, what you might get when you have no oxygen or no photorespiration happening. So we can model that with the model as we did before, and then we can say, okay, here at any given concentration of CO2 inside the chloroplast, we get this value here when there's photorespiration present, and this value here when there's no photorespiration present. So at any given chloroplastic CO2 concentration, we lose out on this much CO2 due to photorespiration. And we can model that very easily, and but unfortunately, that's not necessarily the question we're after. And the reason is that we have CO2 diffusion happening. So the CO2 has to go from the outside of the leaf, has to diffuse through the stomata into the intercellular space, and then onwards into the chloroplast where Rubisco is. So here Rubisco is taking up the CO2. It's also taking up oxygen, producing the photorespiratory pathway, and photorespiratory CO2 is then released in the mitochondria, sort of increasing the CO2 concentration in the chloroplast. And again, this is the CO2 uptake from the carboxylation reaction, so that's the CO2 being taken up here, minus the CO2 coming off photorespiration, that's that CO2, and respiration also coming from the mitochondria. That's sort of our net assimilation rate. So, but diffusion plays in there as well. So we have the CO2 gradient. Because this is a stomata impose a resistance to diffusion, our CO2 concentration in the intercellular space is much smaller than outside. And we can describe this according to Fick's law as the difference between the outside and the inside CO2 concentration is equal to the assimilation rate times this stomatal resistance, RS. So that's the resistance between the outside and the intercellular space. And equally, we get a drop in CO2 concentration from the intercellular space to the chloroplast, also according to Fick's law here. And that's the drop is equal to the assimilation rate times the mesophyll resistance. So what that means is that our chloroplastic CO2 concentration is generally much smaller than, or smaller than our outside CO2 concentration by A times the total resistance here. So we end up with CC being much smaller than CA in general, at least in the light, when photosynthesis is happening. So that has important consequences on when we model photorespiration. So we can sort of graphically display that on our schematic here. So if you take those values here that we measure at a certain chloroplastic CO2 concentration, and we plot our assimilation rate, and then we take our outside the leaf CO2 concentration, CA, and we plot that in here as well. So that line between our chloroplastic CO2, our air CO2 concentration and the assimilation rate at the chloroplastic CO2 concentration is called the supply function. And that slope is equal to 1 over the total resistances. So then we end up with an assimilation rate which has a certain chloroplastic CO2 concentration at our given CA that we measure assimilation rate. So the curious point here is that if we assume that our resistances are staying the same, so stomata are not opening or closing, let's say, then if we take away photorespiration, that means assimilation rate increases, but that also means CC decreases. So as we reduce photorespiration, we also reduce our CO2 concentration and thereby impact our CO2 assimilation. So really, when we take away photorespiration, we increase CO2 uptake by that much, or vice versa. If we have photorespiration present, the plant forgoes about that much CO2 assimilation, rather than this much here, which is assuming that the CO2 concentration in the chloroplast stays constant. Or in other words, where we would ignore diffusion limitation. So if we then assume, well, what happens when stomata close, let's say, well, then the slope changes of that supply function. So as you close your stomata, the supply function would come down. And then as a consequence, the plant misses out less and less on photosynthesis. So the more the stomata are closed, even though photorespiration goes up more and more, what it misses out on, what it forgoes, is becoming smaller and smaller. So we can quantify that and model that in a more robust manner or proper manner. And you can see here, so to the right is when stomata are open, to the left is when you close stomata. So this is stomata fully closed. And here we have the dot dashed line is what you might expect as a typical resistance value in a plant. And you can see as we close stomata, photorespiration goes up. Not by a lot, but it still goes up. On the other hand, the carbon the plant is losing out on is actually going down. So as stomata close and restrict the amount of CO2 going in, the plant is losing or is forgoing less and less CO2 being taken up. And that brings me to my second take-home message, which is photorespiration is not the same as or the CO2 lost from photorespiration is not the same as the CO2 that's the plant forgoes. They're two very different properties. They're related, but they're not the same value. Okay, so how should we then integrate all these effects? And one easy way to do that is, instead of looking at the assimilation rate versus the intercellular or the chloroplastic CO2 concentration, as we've done before, how about we look at the same assimilation rate versus the ambient outside the leaf CO2 concentration. And if we do that, then we include essentially all these diffusive resistances that we may or may not know, but we include them all in this data here. So then we can see, well, in some areas, when we have photorespiration present, we get higher assimilation rates, and in others, we get lower assimilation rates. But we can just take the difference between those two and see what the effect is of photorespiration on CO2 uptake. And this difference here is graphed as a curve down here. So as you can see, with photorespiration, we're losing out on some carbon at low CO2 concentrations, and we're gaining some carbon at high CO2 concentrations. And the crossover point is here where that red dashed line is. And that's very close to the dotted line that I hope you can see in the video. This is the ambient growth condition of those plants. So really the plant is sitting right at that spot where photorespiration is not negative, but also not positive. So we have two areas here. When you go to lower CO2 concentration, then photorespiration becomes carbon negative. And if you were to go to higher CO2 concentration, photorespiration becomes carbon positive. We can also do that with plants measured at different leaf temperatures. So here the solid gray points are, again, 21% oxygen. And the empty points here are 2% oxygen, where we've reduced photorespiration. And you can see that as we go to higher temperatures, the difference becomes bigger and bigger, up to like 35 degrees or so. And again, the transition point between when photorespiration is positive to when photorespiration becomes carbon negative is somewhere around 16 or 17 degrees or so. And only when you go to higher temperatures, like 25, 30 degrees, you see that photorespiration becomes an issue and plant loses out on CO2 assimilation. So what we can then see, what sort of oxygenation rate would a plant want to be at, or to be beneficial? And we can model this oxygenation versus carboxylation rate here based on the data that we've collected down here, and then put our operating point on here. And you can see that up to a point of about a ratio of VO to VC of about 0.3, this is where photorespiration is beneficial. Or similar here, photorespiration is beneficial at low temperature up to a VO over VC of about 0.25 or so. And then again, as you increase your oxygenation rate, it becomes more and more problematic for the plant. But what we can say is that, so a ratio of VO over VC of, let's say, 1 to 4 is beneficial to the plant. So some photorespiration is actually quite beneficial for the plant. So bottom line is, if you look at the assimilation rate with and without photorespiration at a given outside the leaf CO2 concentration, we can integrate both the diffusive effects and the biochemistry in the plant. So the next question is photorespiration good or bad? And is it wasting any carbon? Is it wasteful? And my take on it is, if you look at the flux of carbon coming down on this side here, and compare that to the flux of carbon going up the other way, if this flux here is much bigger than that flux here, then really the plant is not losing or wasting any carbon, because all that carbon ends up in glycine and serine and THF that the plant can actually use and make, so the plant can actually use and would have to come up through other means otherwise. And only if this flux here and that flux here is the same, of course, accounting for that CO2 being lost, this is when I would say, well, maybe the plant is actually losing that CO2 because it's not doing anything productive for, in comparison to the carboxylation rate down here, like the direct pathway. So is photorespiration wasteful? Well, depends, and I think any cost-benefit analysis has to include sort of a detailed view on those fluxes. But what I'd like to point out here as well is that photorespiration has also some other positive benefits. So if most of fixing CO2 requires electrons in the form of NADPH and ATP. So as CO2 is being fixed, light energy can be utilized. So when the plant, let's say, is in a drought condition and closes stomata, it can easily divert flux through the photorespiratory pathway, which also uses electrons and ATP. So it doesn't have to find any way to get rid of those, to get rid of the energy. It just switches to a different pathway. And then when stomata open again, it can switch back to fixing CO2 right away without having to adjust any metabolism. So it's a way to make the plant more resilient, I think, and provide an extra energy sink for the plant under a stressful situation. So third bottom line is, in my view, photorespiration is maybe not all good, but it's for sure not all bad, and has quite a few positive aspects to it. So I wanted to spend a couple of minutes at the end outlining of how we can actually quantify photorespiration in some way. So as I've said before, really what we need to know is the CO2 concentration in the chloroplast and the oxygen concentration, which we assume is constant. So we need to know, in order to measure that somehow, we need to know, well, what the CO2 concentration is on the outside, which we can easily measure, and how big those resistances are. And the first resistance we can measure fairly easy with gas exchange using water, transpired water. But the mesophyll resistance is much more complicated. And we can use one neat tool, which is looking at carbon isotopes. So carbon comes in two flavors of molecule, let's say, a heavy and a light version. And the light version of carbon diffuses much quicker. And it also metabolizes much quicker. So that means the plant preferentially takes up light carbon during photosynthesis and leaves the heavy carbon behind on the outside. And we can measure that as carbon isotope discrimination. And Farquhar et al. About 40 years ago came up with this nice equation here, where they've related this isotope discrimination value that you can measure to what's going on inside the leaf. So we have one component that describes the isotope discrimination observed that's due to this resistance through the stomata. Then one component that describes the isotope discrimination due to the mesophyll resistance. And one component that's related to Rubisco. And then a component that's related to photorespiration and respiration and photorespiration. So this has been one of the main ways, or one of the main ways to assess the chloroplastic CO2 concentration, among other things. And a couple of years ago, I've started playing around with this a bit, and I realized that, well, there might be an issue with that. So if we have our equation here, and all these parameters we can measure, or we need to assume somehow, except for the CC, so then we can solve for CC. But these measures here, B and E and F, we don't really have a clear idea of what those values are. So depending on what value we plug into this equation, we get a much different value for CC out of that. And that's a problem if you really want to estimate your CC accurately. So together with another postdoc in the lab, Misha Holloway Phillips, we designed an experiment how we can test the model itself and see if the model is actually working as intended. And the idea was that if we change the isotopic composition of the measurement gas a little bit, you would assume that it shouldn't affect chloroplastic C2 concentration, or it shouldn't affect the diffusive resistances from the intercellular space to the mesophyll. But when you measure it, it actually does. So here in this graph down here, I've measured GM, which is the inverse of the resistance, the mesophyll resistance. So essentially how hard it is for CO2 to diffuse into the chloroplast against CO2 concentration. And we measured that at with two different tanks that have slightly different isotopic composition. And despite our assumption or our hypothesis that the measured values of GM should be the same, they sort of start to deviate. The lower the CO2 concentrations are, the more deviation you get. And some values even come out negative. So negative conductance, it doesn't really exist, or you'd have to come up with some way to explain that. But in any case, those two values are measured with two different gases are not the same. And that sort of points us to that the model itself doesn't really work under those conditions. And because the composition of the tank only goes into this parameter here, we also have a likely culprit, which is this respiratory parameter down here. So I've rederived this equation again with slightly different assumptions and came up with this equation here. So yeah, it looks very similar to Graham-Farquhar's equation, and I forgive you if you can't pick out the difference. They're essentially the same, except for this parameter down here. So in the old Farquhar et al. Equation, we have RD relative to VC, to the carboxylation rate. And in my new equations, it's relative to the assimilation rate. And this becomes important when you let assimilation rate go towards 0. Then this becomes, goes towards infinite, whereas in the old equation, it stays small. So the actual outcome of those two models becomes very different. So when we take the new model and take the same data and plot this, we get pretty much the same values for mesophyll conductance for those two different gases. And that's really what we're after. So that is an indication that this new model actually is much closer to the truth of what's going on inside the leaf, and a much better way to quantify the chloroplastic CO2 concentration. So now that we have a good way of measuring chloroplastic CO2 concentration, we can have a much better idea of what photorespiration actually does inside the leaf and have a way to quantify that. Okay, and that brings me towards the end of my talk. I'd just like to thank some people who have done the work in the lab and while I was still with Graham Farquhar at ANU, and also some collaborators over the years that have helped me out quite a bit with my work. And then lastly, as you've mentioned before, I've started at the University of Birmingham a little while ago. And while I don't have any funding yet for students or postdocs, I hope that might change in the next little while. So if you're interested, keep your eyes open. Maybe there's something coming up at some point.
Nice. So thank you, Florian. Now we'll open to the questions.
I want to just comment. Can I start with?
Yes, yes. After I will read a question of Professor Eduardo Guzman at the chat.
Okay.
But yes, Homera, you can talk now.
Okay, thank you. First of all, thank you, Florian, for your presentation. It was really nice and really informative for all of us. I am Homera, and I am from Pakistan. So, as you mentioned in your presentation, different kind of things you mentioned that they are responsible for reduction in the photoassimilation rate, photosynthesis. Like it's not just the photorespiration that is responsible solely. So don't just your comments on this. Don't you think this name pho should be renamed for this process?
You mean the VO parameter?
No. As you mentioned, like it's a friend or it's a foe in your
Oh, I see.
topic.
I see what you mean.
So don't you think that the scientific community should rename this term?
Well, I mean, photorespiration is a proper term, I think, because there's still CO2 coming off.
No, Professor. I'm talking about the pho, like the enemy, this term. Because photorespiration is giving us a lot of different compounds.
So my personal opinion is that for many situations, many conditions, photorespiration is very positive and essential for many plants.
Yes.
It's not. I mean, it depends on the condition. So I mean, C4 plants have evolved and they're doing very well under certain conditions, in particular when it's hot. So there's an amount of photorespiration that is not beneficial anymore, and that's where C4 plants, for example, have a big benefit. So but it's not, in my view, it's not that photorespiration is all bad and all plants should convert to C4 plants, because under conditions where temperatures are low or CO2 concentrations are high, plants actually benefit from photorespiration and can compete well against C4 plants. So I think it depends. There's not a good or bad, only good or bad. I think it depends on the condition that the plant is exposed to.
Okay, thank you.
We have a question in the chat from Eduardo, and he is asking, he first said, Florian, thank you for your presentation. Some recent efforts have been made to engineering the C2 photosynthesis to increase CO2 concentration around Rubisco. Do you think this could be feasible, considering that predictions on climate change impacts on crops?
So yeah, I don't know about the feasibility in terms of whether you can actually pull it off with genetic engineering. I'm not a molecular biologist, so I don't know what sort of efforts are involved and if that could be productive. But I mean, certainly the C4 rice project has made good progress, and maybe C2 photosynthesis might be equally successful or beneficial in the end. But in terms of whether it's beneficial or not, I'd have to say the same thing. Depends on the condition you want to grow the plant in, especially if you grow a plant in a hot environment, then reducing photorespiration or increasing CO2 concentration in the chloroplast can be beneficial. But if you grow a plant in the UK in the spring, I think the plant would be much happier with photorespiration than without photorespiration. I hope that answers your question.
Now, Professor Danilo, want to do a question.
Okay, thank you, Luisa. Thanks, Florian, for the nice talk. My question is similar to the Eduardo's question. We have a lot of synthetic biologists trying to or suppress photorespiration or just bypass, like creating new pathways or just moving the substrates or the intermediates of photorespiration back to photosynthesis and so on. So assuming that you could be a synthetic biologist, how would you design a new photorespiration to improve photosynthesis? I mean, what is your feeling about these synthetic biologists' artificial, let's say, pathways that they are trying to build?
So there's two main aspects, I think, that happen at the moment. One is to concentrate CO2 in the chloroplast via a C4 pathway, and that would get rid of much of photorespiration. And I can see that being quite beneficial at high temperatures, let's say. The other one is to have photorespiration still happening, but the photorespiratory release happening in the chloroplast rather than the mitochondria. And you get a little bit of increase in CO2 concentration, which would reduce your photorespiration by a little bit. And that, my understanding is, if I look at the model values coming out, would have only a small effect, or the higher the CO2 concentration you expose the plant to, the smaller the effect would be. So only at low CO2 concentrations, you would actually gain enough to make that worthwhile, would be my guess. So at least on a carbon uptake basis per se, moving photorespiratory release into the chloroplast would only have a minor benefit, would be my guess. I mean, you would rewire all kinds of other biochemistry that the plant has to sort of make up, and I don't know, depending on what you do, it might have a positive or negative effect. But in terms of CO2 uptake or chloroplastic CO2 concentration, it should only have a small effect, I would think.
Thank you.
Now, Ana Carla Lobo want to do a question as well.
Okay, so thank you, Florian. Congratulations for the talk, it was very nice. And my question is also related to Eduardo and Danilo's one. And yeah, I was thinking that we just thinking how to improve and how to delete photorespiration. But now we know for sure that this process is very important and is related with many other pathways in plant metabolism. And I was thinking what to expect in your opinion, considering the climate change and related to the high CO2 concentration that we expect in the future. Because related to your figures and results, we can see that photorespiration is like not that important under high CO2, because we have the difference between low O2 and high O2. Oh yes, O2 is quite, they're quite similar. And also, okay, we know that plants have evolved over the years, and also Rubisco. So we can see how Rubisco properties change over the years when we compare with different species and how they evolved over the years. And also if you believe that there is any strategy to improve photosynthesis and crop yield, considering the climate change as well. So that's it.
So I guess your first question is with this increasing CO2 concentrations. Well, if you double the CO2 concentration in the atmosphere, you would halve the amount of photorespiration. And if you say, well, at a ratio of 1 to 4, oxygenation to carboxylation, you come out carbon negative, then that would sort of double the range of where, or sort of half the range where photorespiration is detrimental as you increase CO2 concentration. But with climate change, it's not just the CO2 concentration, it's also the temperature that increases, and that goes the other way. So higher temperatures means photorespiration goes up. And I guess I'd have to look at exactly what the trade-off is between temperature increase and CO2 increase. But I think photorespiration is going to stay important for quite a ways to come. So your second question was Rubisco or benefits on and how to or ways to improve crop production. Rubisco is like one of the oldest enzymes and has been around, is one of the most important enzymes too. So my guess would be that Rubisco is one of the enzymes that has been optimized most through evolutionary history. So I guess the different people, when you ask them, they give you different opinions. My personal opinion is that Rubisco is actually probably quite well adapted to doing photosynthesis as best as it can. But having said that, we want plants to grow food if we want to modify them. And the way we grow food is not necessarily the same as the way plants grow in a natural environment. So the Rubisco in a crop plant now is maybe not the best Rubisco for that plant that we want to grow in that condition. Right? If you want to grow wheat in Australia, for example, where it's much hotter, we might be able to get a better Rubisco in that wheat growing under those conditions than what is currently in there. So yeah, so I think the plants in general are well adapted to their environments, but because we grow food and maybe outside of the environment the plant is adapted to, we might be able to improve that somewhat via engineering or genetic engineering also.
Now, Fabrício, is you.
Hi, Florian. First of all, I would like to say thank you again for you. Also in the name of Professor Joaquim Albenito, that is not possible to be in today. And my question would be more related with stomato closed conditions. When you have the stomato closed, and it's very common to report this thing related with the increase of photorespiration. And my question is, when you have this stomato closure, you decrease of obviously the CO2, atmospheric CO2 entrance, but you also will decrease the O2, atmospheric O2 entrance. So the O2 for photorespiration would be more related, I believe, I suppose, for the oxygen evolution, in the oxygen evolving in chloroplasts. So my question would be, the first thing is two questions indeed. If the oxygen evolving has been considered to a part of all these modeling projects, because generally we see a lot of things on carbon, on water, but the oxygen evolving is a little bit, I think that is not, sometimes it's not considered, I don't know. And in this case, if O2 come from the thylakoids, the oxygen evolution, other processes that are protecting plants, like for example NPQ, that they will compete with oxygen evolving, if in this case would be prejudicial for photorespiration. So in final terms, could like process like NPQ that avoid the oxygen reactions compete with photorespiration under conditions of stomatal closure?
So in terms of oxygen, your first question is if oxygen evolution would enhance photorespiration. And I think for the most part, the oxygen evolution is not changing the oxygen concentration enough to have any discernible impact on that. So the decrease in CO2 concentration works according to Fick's law. So you get a decrease in absolute concentrations in ppm depending on the conductance and on the flux. So for CO2, that might be a decrease of 200 ppm. So if you have a CO2 concentration of 400 ppm outside, you have a CO2 concentration of 200 ppm on the inside. So you release as much oxygen as you take up CO2. So you would also release so much oxygen that the oxygen gradient is about 200 ppm. So if you have 210,000 ppm on the outside, you would have 200,200 ppm on the inside, which is only a small, very small difference. So really the oxygen evolved is not changing the oxygen concentration in any meaningful way. So really that's what I mean by what's important is the CO2, not really the oxygen. So of course you can experimentally manipulate it by changing the oxygen, but sort of what the plant does by itself is not making a huge difference. So it might be different in C4 plants when you sort of enclose everything in gas-tight compartments. And if you were to evolve CO2 inside the bundle sheath, for example, then in that case, maybe you can build up enough oxygen to make a difference. But otherwise, I think you don't have to worry about the oxygen concentration itself from oxygen evolution.
Thank you.
Now, Professor Milton.
Hi, Florian. Thanks again for being with us. It's a pleasure, and I think we all learn a lot. As I told you earlier, I've been reading your work since a long time, and I'm really enthusiastic on your field. And I think with this opportunity, we can collaborate in the near future. So I have two questions. One is regarding what Fabrizio already said, is the importance of the balance of losing carbon and the photoprotective mechanisms as an electron sink in plants exposed to high temperatures and excess light. There's a common issue here in the tropical plants in Brazil or tropical areas. And the other one is regarding the difficulties of measurement and photorespiration by HCR curves and modeling. If you can talk more about those difficulties that you can find and the problems that can make on our results and assumptions. And thanks again for being with us.
Sorry, the first question was?
The first question was regarding the balance between losing carbon and the photoprotective mechanisms of avoiding photoinhibition and excess light.
Yeah, that's a very good question. And unfortunately, I don't really have an answer to that. I mean, we know that there is this benefit, but it's very hard to quantify that. And I haven't figured out a way how to actually properly do that. Because it's sort of, I think what you have to do is you have to look at least in an evolutionary sense, why photorespiration exists, is you have to look at long-term history. If you have a plant that only needs photorespiration as photoprotection every 1,000 years, and if you don't have it, you die, then, well, over the long term, you still go extinct if you don't have it. And you might lose out on a lot of carbon in those 1,000 years in between, but it becomes very beneficial when such an event comes around. So yeah, I think it's very hard to weigh out the carbon loss versus the benefit from making a plant resilient. But that doesn't mean it's not an important aspect to have a resilient plant that actually can withstand any sort of adverse environment that it might experience throughout its evolutionary history. So yeah, I don't know how to quantify that, but I'm fairly certain that it does have some benefits that might come in important at some point at least. In terms of crop plants, we might be able to decrease the resilience towards and put more towards carbon gain if we can actually make sure the plant is not experiencing that stress. If you have a plant grown in a glasshouse, well watered, you might not have to worry about stomata being closed or having too much light or any of that. So maybe you can sacrifice some of the resilience and get more benefit from that. But yeah, I don't know how to properly quantify benefit and cost in that sense. Does that answer your question?
Yeah, sure. And yeah, I think that's the issue when you always think about crops and plants from glass house, and we will open that for the biodiversity that we can find here in tropical areas, anything like that. And as I told you, it's my kind of the field, the photoprotector mechanism, and things like that. So that's my my interest, and I think we have a lot of studies and questions. I think primary questions, then answers that we need to ask regarding the photoprotector mechanism of photorespiration.
But I mean, maybe one way you could address this is if you look at plants that have evolved in a very highly variable environment, and compare that to plants that have evolved in a very stable environment. Do you see any evolved trade-offs between the photoprotective mechanisms and being able to grow as fast as you can? And you might find sort of a general trend.
Yeah, and the second question that I make to you is regarding the problem that we can face trying to measure photorespiration by modeling ACI curves. What are common issues and problems that we can find that we can compromise on our results and things like that? Misinterpretation, I think. Yeah,
so I think you can measure it easily and not worry about modeling. So if you measure at a common ambient CO2 concentration, measure your assimilation rate with and without photorespiration, you can get a very accurate impact on the photorespiration on assimilation rate. But unless you know your chloroplastic CO2 concentration, you can't model it easily and extend it to other conditions. So for that, you actually have to know all these components, the CO2 diffusion into the chloroplast. And I think carbon isotope discrimination is a very neat way to get at those parameters. So if you want to model it to conditions you haven't measured, then you need those accurate measurements of CC. But if you're just interested in the effect, you don't need to know any of that. You just measure with and without photorespiration at a given condition, and you get a good impact or a good quantitative effect of photorespiration on assimilation rate.
Okay, thank you very much, Florian, for being with us again. It's a pleasure.
Professor Danilo has another question.
It's exactly about this method that you have improved the equation to estimate mesophyll conductance by isotope discrimination. My question is, given that C3 and C4 plants has different discrimination based on Rubisco and Pepsi activities, shall we have one equation for C3, one equation for C4, or this is global? We can use, but taking care of about the interpretation.
Yeah, unfortunately, the equation I've shown you is only for C3 plants. And the situation is much more complicated in C4 plants, and you don't really capture what's happening in C4 plants with that equation. There is an equation for C4 plants, and like an old equation also Farquhar et al. Who, and as well as Susanne von Kammerer and Bob Furbank came up with an equation a few years after that. But those equations have the same issues as the Farquhar et al. Equation had in C3 plants. So I'm currently working on trying to address that in some way. But it's much more complicated, so you can't use the same equations for all cases. C2 plants is another issue that you can't really just use one of those equations and think you end up with the right answer.
Yeah, just
makes things tricky.
I just remember now that, for example, Geomflexes is also trying to estimate. We are also doing this ferns and bryophytes and so on. So I guess this is even more complicated, right? To estimate based on isotope discrimination or gas exchange analysis. We don't have any equation for these basal planes, right?
Yeah, so if you have a C3 plant like gymnosperms or probably ferns, the equation should also work. I mean, if you don't have stomata, for example, you might have to get rid of that stomatal component of it. But the main parameter driving all this is the Rubisco component, and that is sort of universal between all C3 plants. In C4 plants, you have the problem that Rubisco component is hidden in the bundle sheath, so you don't really measure that. What you measure is mostly the PEP carboxylase component, so that's what makes it different. But if you have Rubisco as the primary fixing enzyme, in principle, you can use the C3 equation, I think.
Good, thank you.
Florian, I have a question more or less similar with Danilo said in the end. Is more at evolutionary point of this issue, because I know that the most of data that we have is related with Arabidopsis because of our limitation to study with mutants and to do these computational models. I want to know if we know, because I believe that everything that you see and you find in nature is there because, and sometimes is good for the plant. You have advantage, yes? And the first question is, in your opinion, there is some reason to use 3 compartments in the cell to do the photorespiration, or is just because of different spatial distribution of the enzymes? It's a simple question, but I never see any.
No, it's a very good question, and I don't have an answer for that. There might be some biochemist who's more an expert on why that is happening that way. But you have the conversion of glycolate to glyoxylate produces hydrogen peroxide, and that's for one toxic or highly reactive, and also a signal molecule. So there might be a reason why you want that sort of separate from the cytosol or the chloroplast, and you have it in this compartment where it doesn't really do much harm, or where you can separate the signal. So I don't know if that's the reason, but I can think that something like that might be a good reason why it is happening in different components like that.
Yes, and the other question is, do you have some data related comparing more primitive plants with more complex, the rate of the affinity of Rubisco changing along the time with the evolution of O2, showing that with the higher concentration of O2 during the Earth history, so along a million years, the affinity of O2 was increasing or decreasing. Do we have this data comparing different plants?
So we do have Rubisco kinetic parameters for all kinds of different lineages, and they do change. So when you look at plants growing at the moment and under ambient concentrations, you can see, for example, C3 plants have a much lower CO2 concentration in the chloroplast than C4 plants. So C4 plants operate at a few thousand ppm CO2 in the bundle sheath, and C4 plants have much different kinetic parameters for Rubisco than C3 plants. So the C4 plant Rubiscos are generally much less specific for CO2, but on the other hand, fix CO2 much faster. So there's this trade-off between specificity and speed. And similarly, you have algae that have CO2 concentrating mechanism that have different kinetic constants. So you get a wide range in parameters, and often there's a trade-off between the speed Rubisco operates in and the specificity. But that trade-off is not set in stone. So even though there is a trade-off, there seem to be some Rubiscos that sort of can have both or neither.
So all the Rubiscos that we know have these two activities. We don't have any Rubisco with just carboxylation activity, yeah? All of them.
As far as I know, we don't, no?
Yes. Okay.
I mean, CO2 and oxygen look very similar on a sort of molecular basis. They're all small molecules with two oxygens, and it seems to be very hard to distinguish. Either that, or there's a high selective pressure for photorespiration to be present. And it seems to be good to have that tied to your carboxylation at a certain rate. So I think those are the two options.
Yeah, because unfortunately we can measure enzymatic activity on fossils or plants that don't exist anymore. But maybe we can find a domain on the genetic sequence responsible for the oxygenation activity. I don't know if this study exists, and maybe compare primitive plants and show exactly moments when the photorespiration starts, or to prove that this process always exists since the beginning. Maybe it will be a curious process.
I think the general assumption is that Rubisco was always sensitive to oxygen, yeah. And it just changed how sensitive and what the specificity was. But I'm not sure if you can go to other lineages and sort of extrapolate, look at the kinetics and extrapolate what those kinetics would have been 3 billion years ago when CO2 was much higher. Because, I mean, even those lineages are primitive, they still have like all those millions and millions of years of evolution behind them. Yes. So just because they're a primitive plant doesn't mean the Rubisco is still the same. But I don't know if there's any way to sort of estimate what kinetics would have been a long time ago.
That's true. Thank you. And Fabrizio, you have another one.
Yes, thank you. My other question that I have is about a topic that you mentioned briefly in the presentation about that phase of the curve, the triose limitation, the final phase. I heard some polemics recently about that, and if this would be an issue on field conditions, natural conditions or not. I would like to hear your opinion about that. Yes, this could occur only in high CO2 concentrations, or maybe if some environmental factor could affect this triose phosphate limitation, in your opinion.
Well, I think in general, it's as important as electron transport limitation, maybe even more important. But pretty much, if any plant I've looked at and I want to measure TPU limitation, I can't find it. So it might have to mean you have to go to lower temperatures or to higher CO2. But if you find the right condition, you can always find it pretty much. So I haven't found any plant yet where you can't find it. I mean, it becomes maybe more or less important depending on what you acclimate your plant to and what you measure your plant at under which condition. But yeah, if you, I think it's just as important as Rubisco limitation or electron transport limitation, if that makes sense.
And about nitrogen limitation conditions, could that be an issue for TPU?
Well, nitrogen limitation would affect all of that essentially, right? So it, really, as a plant, if you acclimate to your environment perfectly, you want to be equally limited by Rubisco and electron transport limitation and TPU limitation. So that's when you get the most flexibility. And so if you're limited to your environment perfectly, independent of how much nitrogen or how much phosphate you have, you'll always be co-limited by all three limitations. And only if you're not perfectly acclimated to your condition, if you say you grow a plant at high temperatures and then you shift it to low temperatures, then you can see one limitation becoming more dominant over the others under that new condition. But yeah, if you deprive nitrogen or if you add extra nitrogen, you shift that plant out of that acclimation for a little bit, but ultimately, in the long term, I think you should still be equally limited by all three limitations. And in that sense, TPU should be just as important as the other two limitations.
Thank you. Thank you.
So does anyone have more questions to Florian? It's now or I don't say never. It's now or just after. So anyone more? So we have a very, very nice discussion, Florian. I believe that this part is better than other presentation because you can share very doubts and about this hot topic. So to finish, I would like to thank you again for giving for us your time.
Thank you for having me, and I'm glad I got so many questions. That means you actually managed to pay attention to what I was saying.
It was not sleeping. So yeah, so thank you everybody for participating of our lab talks, and I hope that you enjoy and see you in the next topics. Okay.
Okay. Yeah, I hope I might see some of you in person at some point.
Yes, I hope you keep in contact to collaborate and still doing science together.
Yeah, sounds good. Anytime.
Okay.
All right. Thanks very much.
Bye
Thanks,
bye.
Bye.
Bye bye.